This would be complicated once in 5 years or so.
Okay, couple of heads up. If you have anything in the incubator, before you leave, it should go in the refrigerator. And don't leave them in the incubator, so the incubator should be cleared out by this afternoon. At just a few minutes after 11, I will have your exams for you in your lab drawers. And so you can come by and pick those up, and I'll send out some information about class stats and things like that. But I've just got to get some grades recorded, so those will be ready. We do have a lab exam when you come back. The lab exam is over really just 4 or 3 labs, 14, 15, and 16. But there's a fair amount of information in there. I'll double check. I think I have a study guide for that lab exam. And if I do, I will send that out to you this afternoon. Pay attention to the questions at the end of each lab. And a lot of this one is more about interpretation of results. Okay? So if a tube turns yellow, do you know what that means? Some of this involves a little bit more understanding the theory and the chemistry of the reactions. So it's not quite as much hands-on stuff as it was before, and a little bit more interpretation and theory type things. So any questions? Almost there, folks. No? She says no. Okay, so what we're doing right now as we kind of start marching our way through the third line of immunity is before we put everything together and start getting into some fairly complicated diagrams that shows all of the interactions that can take place between antigen-presenting cells, B lymphocytes, T lymphocytes, various cytokines that are produced. We need to give you enough background so you can begin to understand the complexity of those particular interactions. So where we started with last time was said receptors are really important because receptors are involved in, number one, recognizing what is foreign versus what is self. And that's critical in making sure we have an immune response that is selective. But receptors play more than just that role of recognition. One of the other things that receptors do is they bind to various cytokines. And when that happens, the binding of the cytokine to the receptor initiates a signal transduction cascade. And basically, binding outside the cell influences activities inside the cell. How does it do that? Well, ultimately, it is affecting gene expression. It might be gene expression of transcription factors that affect other genes, or it might be genes that are affected firsthand. But after those receptors are stimulated by a cytokine, the cell starts doing something different. One of the things that it can do differently is start going down a different developmental pathway. And so naive B lymphocytes, for example, start differentiating down a pathway of becoming plasma cells so they can produce antibody, or becoming memory cells so they can then respond to the infection next time. T lymphocytes start diverging down different pathways following cytokine stimulation to become either a T helper cell, or a T killer cell, or a T suppressor cell. And so that binding of receptors to the cytokines really does influence the differentiation pathway of the cell. Okay, one of the first types of receptors that we talked about, and the only one we've talked about so far, are the MHC receptors. Remember, MHC stands for multi-histocompatibility. They were discovered in the context of, sometimes you transplant tissue and it takes, and other times the transplanted tissue is rejected. Well, what's going on? Well, as it turns out, each one of your cells has kind of a unique signature. And that unique signature is a bunch of molecules, many of which are glycoproteins, on the surface of your cells, and they basically kind of give those cells identity and say they belong to Luke. So if we then transplant tissue, and since those cells belong to Luke, hopefully as the immune system is developing, immune cells that would respond to self cells have been eliminated. One of the things we talked about is that the immune system has tolerance. It has tolerance for your own tissues and shouldn't respond. So that deletion of cells that respond to your own self-antigens is a really important part of the development of your immune system. And sometimes it goes awry. And if that happens, then we can have an autoimmune disease. So you have this cell signature on the surface of your cells. And that's really important because one of the other things we said that characterize the immune system is surveillance. And you got all these mobile macrophages and neutrophils migrating around within your tissue, checking out cell surfaces, that group growth that we referred to, and seeing if things belong or don't belong. And one of the things they're looking for are these MHCs and molecules associated with the MHCs. That's a big part of your cell signature. And if those macrophages and neutrophils determine this cell belongs to the organism, they should leave it alone. But if they decide this cell doesn't belong to the organism, okay, let's respond. Let's do something about this invader. Where do we find class 1 MHCs? What kind of cells? All cells except blood cells, okay? Where do we find class 2 MHCs? Anything that could be potentially an antigen-presenting cell. Okay? What we're going to see later on when we talk about the T cell response is that there is some restriction going on in terms of T cells and what kind of cells they can interact with in the body. There are some that are class 1 MHC restricted, and there are other types of T cells that are class 2 MHC restricted. So they'll only interact with cells if they have the proper MHC on their surface. So I also showed you this diagram right here. And this shows you the endogenous processing of antigens that ultimately end up on the surface of cells associated with an MHC1 receptor. So here's a typical cell in your body. It's producing all these proteins. Those proteins turn over, and pieces of those proteins end up getting stuck onto MHC1 receptors inside the cell. And then that combination of self-antigen and MHC1 receptor gets packaged up into a vesicle and eventually becomes inserted on the outside of that cell's membrane, giving it that cell surface signature. So normal cells are studded with MHC1 receptors, and nestled into that receptor, remember they've got this little groove right here, are pieces of your own antigens. So this basically says to cells that are surveilling your body, this belongs. Okay? Well, what if this cell becomes damaged in some way? It becomes virus infected or cancerous. Then it starts producing different proteins. It starts producing cancer proteins or viral proteins. And those end up getting displayed on the surface of cells. So your signature changes. So now those surveillance cells are going around and they go, whoa, wait a minute. I see an MHC1 antigen complex. It doesn't belong. I'm going to respond. Okay? MHC2s are the result of exogenous antigens becoming complex with those receptors. So, if I've got an antigen-presenting cell, a phagocytic cell, and I've got a bacterium with its unique antigens, its PAMs, then that macrophage can recognize those PAMs by the toll-like receptors. That stimulates phagocytosis. And then eventually that pathogen is internalized. It's degraded within the phagolysosome. But then pieces of that internalized pathogen become associated with those MHC2 receptors in the cell. They hook up together, and then they're transported out onto the surface of the cell. So now these antigen-presenting cells, in addition to having MHC1s with their own antigens, have MHC2s with foreign antigens on them. And they then will show those antigens to other cells, like a B lymphocyte or a T lymphocyte. So if you're an antigen-presenting cell, you've got to be able to do this. This is how you show or present the foreign antigen. Now, that concept's really important. We'll be coming back to that concept. There are another set of receptors that are found on the surface of immune cells. There are a whole bunch of types of receptors. We're only going to be talking about a few of them. I mean, the number of receptors that are on the surface of immune cells are just crazy complicated. But there's another class of receptors that are found on immune cells that are called CD receptors. And here's a list of some of the types of CD receptors that are found on various players in the immune system. So B cells, T helpers, T killers or cytotoxic, T regulatory, also that's called suppressor. Another wrinkle you haven't seen quite yet, we'll talk about later, is there are actually two types of T helper cells. There's T helper 1 and T helper 2. But here are some of the representative cell surface receptors. Okay? CD40, CD4, CCR5, CD4 again, CCR3, CCR4. There's this huge list of these. I mean, what does this imply to you? There's been at least 40 of these CD receptors identified and characterized. But there's two of them that I just want to point out to you that are important. Okay? And they are CD4 and CD8. CD8 are found on the surface of C cytotoxic cells, or the killer cells. CD4s are found on the other T cells, the helper 1s, helper 2s, and suppressors. And the only reason I'm telling you about this is that sometimes in literature, you will see reference to a cell being referred to as not a killer or a helper, but instead a CD4 cell or a CD8 cell. And that was the case in your seminar, right? They were talking about CD8s. Yep. So I just wanted you to be aware of that. So this is another receptor, and we'll see some of these receptors, these CD receptors, in diagrams later on when we start looking at receptor matching between antigen-presenting cells and B and T lymphocytes. So I just wanted you to be aware of their existence and what those designations refer to. This also shows you something we'll talk about a little bit later on. What do some of these particular cells secrete? Okay, well, B cells secrete antibodies. But in order to secrete the antibody, you need a proper signal. Okay, and sometimes those signals are perceived by these particular receptors. T helper cells can produce various interleukins. We'll talk about those in a minute. Interferons. Cytotoxic cells produce these enzymes called perforin and granzyme. We haven't talked about those yet. Regulatory cells produce cytokines, including interleukin 10. So I think it's important to understand that these white blood cells right here are typically going to be secreting some kind of a lymphokine. And in order to do that, they've got to be stimulated by signal 1 binding their antigen that they're destined to interact with. But more often than not, usually, some kind of a cytokine which will react with these some of these kind of receptors. So that's just context for you right now. Okay, a couple of really important receptors we need to dig into are the B cell receptors and the T cell receptors. And these are the ones that react with antigen. And as it turns out, MHC receptors as well. We'll dig into that a little bit more in just a second. I'm going to spend most of my time talking about the B cell receptor. and then we'll show you the T cell receptor and show you how it differs a little bit. So, here is a model of a B cell receptor. And for right now, you can disregard these little globular proteins right here, and instead concentrate on just this part right here. So, what are these little lines? They represent polypeptide chains. And you'll notice that the B cell receptor, this half is a mirror image of this half. So we say that that B cell receptor is a dimer. Something else that you'll notice is that the two chains are different lengths. This one has more amino acids in it than this one does. We designate those particular chains heavy chains, the smaller ones being light chains. Okay, makes sense? And notice that they are joined together by disulfide bonds. The amino acid composition of this light chain is identical to the amino acid composition of this light chain. Likewise, the amino acid composition of this heavy chain over here is identical to this one. Now, I said that these particular receptors are critical because they're involved in recognizing foreign antigen. So where does that binding actually take place? It takes place in this region right here and in this region right here. So this is considered to be the antigen binding site. So if this part of the dimer binds antigen number 1, what does this part bind? Antigen number 1. Okay, they're going to have identical antigen binding sites. So these B cell receptors bind two molecules of the same antigen. So you're not going to have one antigen binding over on the left side over here and a different one on the right side. Two molecules of the identical antigen. And that's going to be important later on. It's not quite so important right now for our content, so that's going to be important later on. Okay, if we look at these particular light chains right here, there are two types of light chains, and they are designated kappa and lambda. In some B cell receptors, both of them are kappa chains. In other B cell receptors, they're both lambda. But we never mix a kappa and a lambda. Okay? So it's either two of one or two of the other. And these can differ in amino acid sequence. Heavy chains consist of kind of two modules or two building blocks that are designated V and C. V stands for variable and C stands for constant. The terms variable and constant refer to amino acid sequence. The variable regions tend to be up here at the end of this polypeptide chain, up here before the bend. And then the constant tends to be this region right down here. So this tends to be the constant region, and this tends to be the variable region. In receptors, there can be 5 different types of constant chains. And we're going to get into that a little bit better later. There are different classes of receptors, and there are also different classes of antibodies. What's nice about this particular discussion is once we talk about the structure of the B-cell receptor, you have learned then, de facto, what the structure of an antibody is. So there are 5 different classes of receptors and also 5 different classes of antibody. That's determined by what your constant region is. Okay. So for example, there is a class M type of antibody. All antibodies that belong to class M have the same constant region, same sequence of amino acids down here. But each different antibody within that class has a different sequence up here in the variable region. This idea that the receptors are built out of blocks or modules is going to be really important in a minute. Okay, so 2 heavy chains, 2 light chains, the ability to bind 2 molecules of identical antigen are some of the major characteristics of this B-cell receptor. This is a T-cell receptor, and it looks a little bit different. And that's pretty much all I'm going to want you to know about the T-cell receptor. It doesn't have 2 heavy chains and 2 light chains. It's really just got these heavy chains. But notice how clustered around that T-cell receptor are some CD receptors. That's going to be important later on. We'll come back to that later on. So this would be the antigen binding site up here for the T-cell receptor. And notice there's only 1 antigen binding site, not 2. So on the T-cell, the T-cell receptor forms a functional complex with these CD receptors. So this is referred to as a T-cell receptor CD complex. You don't see that on the B-cell receptor, okay? You don't see that association with the CDs. Okay. So, let's look at the B-cell receptor for a second. Remember that those particular B cells are born in the bone marrow and then become educated in the bone marrow. What does that education process involve? It involves building a receptor, synthesizing proteins in your cytoplasm, and then transporting those out into the cell surface and putting together the right heavy chains and light chains to make that B-cell receptor. And remember that B-cell receptors have this unique antigen binding site here. The nature of that antigen binding site is a consequence of what's the sequence of amino acids in these variable regions of the light and heavy chain. This defines the shape of those binding sites and what antigens they can bind. What's the relationship between a receptor and an antigen? Very specific. A particular receptor only binds one antigen, just like enzyme substrate specificity. So during that education process, we have to build receptors that will interact with potential antigens. And then once that education has happened, remember then the B lymphocytes that are now educated but still immature or naive migrate out into the lymph nodes or the spleen or whatever and sit there and wait to encounter their antigen. How many antigens are there out there in the world that B cells would be expected to interact with? Billions. I mentioned last time that we can even get an immune response to a brand new novel chemical that's been produced in the lab. And we'll get a specific antibody produced in response to that new antigen. So B lymphocytes, and the same thing applies for T lymphocytes, have this expectation that there will be receptors that will interact with any possible antigen that could exist. It's a pretty tall order. So one of the questions becomes is, how many different types of B lymphocytes develop and become educated within the bone marrow? Well, could it be just a single type of B lymphocyte? No, because each B lymphocyte would have to have on its surface 1 billion different types of receptors. There's not room for that. And remember that I told you earlier on that each B lymphocyte actually has about 50,000 copies of the identical B cell receptor. So we need more than one cell. We need a different lymphocyte with a different receptor for every possible antigen. It's conservatively estimated that there could be 10 to the 23rd power different antigens that B cells would likely have to interact with. Man, so we've got these cells that are being born in the bone marrow. They're coming from stem cells. So stem cells start dividing and differentiating into these different B lymphocytes. Potentially 10 to the 23rd power different types, each of which has a unique receptor. Well, what are receptors? They're polypeptide chains. What are polypeptide chains coded for by? Genes, right? How many genes would you need to have to build 10 to the 23rd power different types of receptors? A lot. How many genes are there in the human genome? Do we know an estimate now? Current estimates indicate probably about 20,000 genes in the human genome. That's not enough genes, even if every gene in your stem cells coded for just receptor proteins to even begin to get near the number of receptors we needed to make. There's a real disconnect there. Something really bizarre happens in the bone marrow to these cells that start going down this differentiation pathway toward forming B and T lymphocytes. Something really interesting happens within their genome. A phenomena called gene shuffling occurs. There's an enzyme that's really important in this gene shuffling process called RAG. RAG stands for recombination activating gene protein. And within the genome, there are sequences of nucleotides called RSS sequences. Recombination signal sequences. And basically what this RAG enzyme does during B and T cell lymphocyte development is makes cuts in the genes that code for these receptors. Okay, so these are going to be receptor sequences, receptor genes. Cuts them up and then reshuffles them and rejoins them in different orders to make new combinations. How many of these receptor genes are there in totality? Approximately 200 genes encode receptor genes. And you go, man, 200 genes? How can 200 genes encode enough information to make billions of different receptors? Because of this reshuffling of that genetic information that takes place during that cellular development. Okay, heavy and light chains are built out of gene segments or modules. Heavy chains are made out of 4 gene segments. V, D, J, and C. V stands for variable. D stands for diversity. J stands for joining. And C stands for constant. To build a heavy chain of a B cell receptor, what needs to happen is this RAG enzyme has got to cut out and grab one variable module, one diversity module, one joining module, and then stick that together with a constant module. And that makes that heavy polypeptide chain. Now, there are several variable modules. There are several diversity modules. There are several joining modules. So you can start to see that we're getting some combinations here that can occur that would give us some significant different number of receptors. Light chains are made up of 3 modules. The V, the J, and the C module. There's no diversity module involved in the sequence of light chains. So if you want to, think of these particular little modules right here that RAG recognizes because there are proper RSS sequences bordering them, and cuts them out and joins them together. Think of this as a deck of cards. And what RAG does is it deals hands of cards. You're going to play 5-card draw. 5 cards will be pulled out of that deck of 52 cards. They're randomly shuffled, and then those hands are dealt out. And you can get a bunch of different combination of hands. RAG shuffles the genome by cutting out a V sequence, a D, a J, and a C, and joining them together so that then this particular message can be translated into a protein, and that protein can then be packaged up and inserted in the membrane as part of a receptor molecule, okay? The heavy chain portion. Remember light chains, we said that there are 2 types of light chains. There are light chain lambda and light chain kappa. They have different variable regions. They have different joining regions. And in order to make a light chain, you need to put together one of the V's with one of the J's with one of the C's. So what this particular chart does for you is it just starts showing you numbers of possible combinations. For example, just given this genetic information, and this doesn't count the constant regions right here, this counts just the V and J and C, D regions, we can get 120 different combinations of light lambda chains. We can get 200 different combinations of light kappa chains. And when you build a receptor, any lambda can associate with any heavy chain. For heavy chains, there are 100 V regions, 27 D, and 6 J, and so that gives you 16,200 different combinations. And then when we start putting these together, a heavy with one of these or one of these, we're up to about 5 times 10 to the 6 different combinations. That's a lot. But it's not quite where we need to be. So there are limitations in terms of how many different combinations gene shuffling alone can generate. But it's pretty impressive, the number that you can get right here. So where do we get that additional diversity that we need?
On the slide before, how you showed the D, and J coming together, everything sometimes works sequences we have to have that as well.
They're only showing you transcription of the variable portion of the heavy chain. They're not showing you transcription of the constant portion. So there'll be another set of C genes over here, and one of those 5 C modules would be picked off and associated with this. So they're just not showing you the whole set of combinations. They're only showing you the variable region of this portion right up here of the heavy chain. Okay? So where do we get that additional diversity that we need? Well, this enzyme RAG comes into play again. RAG can go in and randomly remove portions of the V and D or J. Sorry, D and J segments. Before joining together. So normally, that RAG enzyme will recognize particular boundaries for D heavy 1 and D heavy 2 and splice those out properly. But after it does that, it can go back in here and go, I'm going to take out another amino acid. I'm just going to chop another one off. Or I'm going to chop another one off over from this J signal, which alters your amino acid sequence. And because it's random, we get another whole set of possible set of combination. There's another enzyme that randomly adds nucleotides. To the VDJ segment. So once it's formed, once this is all stuck together by RAG, another enzyme comes in, and I don't have the name of it. I don't know what that is. I'll look that up. And maybe adds a few random amino acids here and there to the ends. That gives you new combinations, new source of diversity. Another thing that happens is that these genes that encode the variable regions tend to hypermutate. They tend to become very, very kind of unstable and randomly mutate. So if the nucleotide sequences randomly change within VH1, that changes amino acid sequence. One of the things that we kind of undersell to you in cell biology, definitely, maybe less so in genetics, is this idea that your genome is really stable and kind of fixed. That certainly is not true within the stem cells of the bone marrow that receive the proper signals to start differentiating down these lymphocyte pathways. Something happens within the genome of those cells that make them highly unstable. And all of a sudden they start snipping up certain regions of their genome and recombining them to form all these different combinations. Other enzymes start adding random amino acids to those gene products once they've been translated into protein. And mutation just accelerates rapidly in some regions of the genome, particularly that encode these variable regions of the heavy chain. All of these discoveries about the genetics of receptor diversity and the development of B and T cell receptors just blew people's minds when they discovered it. And for a long time, people just go, no, that's really not happening. And some of the early researchers who were involved in these particular types of experiments had a really difficult time selling their colleagues about the fact that the genetics of these particular B and T lymphocytes as they were developing were very different than the genetics of the other somatic cells of your body. And that was really hard to accept. I don't know how many of you have heard the story of, did she talk about Barbara McClintock and genetics and the jumping genes? Yeah, I mean, McClintock was just ridiculed for years because she had this concept of these transposable elements, these genes that moved around, and I mean, everybody just said she was crazy. It took a long time for people to appreciate how insightful she really was. It's a very similar kind of thing here with this combinations of different receptors that can form. You put all these things together, the gene shuffling and some of these other factors, and now we start basically being able to form an unlimited number of receptors by just random recombination. Now, one of the things that's going to happen is that likely many of those receptors will be matches for self-antigens. And so once those cells with particular receptors are generated, we have to get rid of them. And so learning to tolerate your own antigens is a particularly important part of your cell's immune response. So here's receptors being translated into messages Those messages are then, transcribed into messages. Those messages are translated into proteins, and then the proteins move through the endoplasmic reticulum. Some carbohydrate is added to them, and eventually those receptors accumulate on the surface of the cells and then sit out there and wait for particular antigens. I'm going to talk about that later. That's not a relevant slide right now. So this shows you some possibilities for different types of receptors that could be generated. And hopefully those that interact with your self-antigens will be destroyed. They'll go through a process of apoptosis. And only cells that recognize foreign antigens will be allowed to survive. And this type of clonal deletion occurs in both T cell receptors and B cell receptors. So we need to get rid of both B and T lymphocytes that would react with self and only allow ones that react with foreign to survive. Okay, that's a good place to stop. So have a good break. Get some rest. Get some turkey. Because it's going to be pretty busy the last couple of weeks. And don't forget to take